Good morning, all. Welcome to my presentation. Today, I'm going to introduce to you a resource that allows us to comprehensively screen for drug and drug metabolites in untargeted metabolomics dataset. This resource was developed through support from the mPrint Consortium as well as other funding sources. So I would like to give an acknowledgement to mPrint at the beginning of my presentation. So we know that chemical exposure have a profound effect on the health and disease of our body. Among the chemical coming from food, from environmental pollution, from the microbiome, drugs represented an important component of the chemical exposure that have dramatic effects on the biology of our body. So because of this, I actually get a lot of requests about drug analysis in my daily life as a metabolomics scientist. So one of my collaborator, of course, with high relevance to the imprint, came to me and she asked me, well, Nina, can you use your magic on targeted metabolomics and help me to detect all the antidepressants in my milk samples? So what I did is basically analyzing the samples on the mass spectrometry in a untargeted fashion, and then I compared all the MS spectra recorded on the machine with a library of spectra of known compounds. So what I get out of this library match is a long list of chemical names like this. There are definitely simple names such as caffeine that I can immediately understand, but there are also a lot of really complex chemical names that I have no idea what they are. Seeing those complex chemical name as well as the name of some drugs that I'm not familiar, it's literally not no different as seeing those emojis down there, right? So basically, the magic non-targeted metabolomics I do in my daily life was basically to Google about all these chemicals one by one. So after I spent a lot of time trying to figure out what are these chemicals and where do they come from, I finally was able to get a short list of antidepressants I detected in these milk samples back to my collaborator. So the collaborator was definitely happy with the output that she was able to know the antidepressants in the milk sample. However, she starts to tell other people that Nina can analyze drugs in any samples, right? So the second collaborator came to me, and this time he wants to understand all the antibiotics in the fecal sample because he cares about the gut microbiome. So I basically did the same analysis again, except this time I spent even longer time to annotate to figure out all the name of the chemical. And finally, I was able to get a short list of antibiotics I saw in the fecal sample back to my collaborator. But of course, the story doesn't stop here, right? When my third collaborator came to me and want to understand the illicit drugs in wastewater, I was like, hold on. So I really love science, I love chemicals, but I also don't want to spend the rest of my career just Google about chemicals. So all this request about drug analysis really motivated me to think, can I actually develop a one-stop shop for drug analysis? The idea is that I want to develop a spectral library specifically for drugs, and also, I want to connect pharmacologic metadata to all the entries in my drug library. So the hope is that with this resource, whenever I get a sample, no matter if it's wastewater, breast milk, or fecal sample, I can immediately annotate the non-target metabolomics data using my drug library, and within one click to connect with my drug metadata, I will be able to tell you all the antibiotics, all the antidepressants, as well as the function of all the drugs that I saw in the samples. So this is where I started with this project, and the first step was to develop a reference library for drugs. So where do I get reference spectra? So first, I mined public MS library, which include both the GNPS mass bank as well as most of the common open access open access MS libraries resources. So in addition to that, we also collaborated with Dr. Karina Brax from Czech Republic because Karina recently analyzed more than 2,000 chemical standards on the mass spectrometry to generate their reference spectra. We also collaborated with Dr. Li Binxu, who incubated more than 2,000 drugs using in vitro liver cultures trying to capture the reference spectra for drug and their metabolites. So in total, through this comprehensive reference collection efforts, we were able to collect more than 100,000 reference MS2 spectra that covers about 5,000 drugs in our resources. So as I promised you earlier, when you were using this drug library, you will no longer need to Google about drug anymore, right? So to achieve that, originally from the beginning, I collected pharmacologic information about the drugs from online drug knowledge base that include the FDA Pharmacologic Classification, Drug Bank, Drug Central, Campbell, and the Broad Institute Drug Repurposing Hub. So we documented the therapeutic area, mechanism of action, as well as other information that's not shown on this figure for all of the drugs within the drug library. However, when I showed this resource to a pharmacist in our lab, Dr. Kine, she's told me that she actually noticed a bunch of inconsistency and wrong information in these pharmacologic data collected online. So Kine actually ended up going through the metadata in the drug library one by one manually to make sure that they were correct and comprehensive to the extent possible. So I want to give a big shout out to Kine. She's really the hero of this project. However, when we use this resource, we notice another problem. We notice that there's not only drug, but also some of the endogenous molecule included in our drug library. This is because these endogenous or food-derived molecule can sometimes also be used as a drug. So for example, cholic acid, the bile acids can be used for liver disease. Hormones, there are hormones used for endocrine disease, and there's even a food sweetener included in the drug library because it can also be used as a laxative. So in order to differentiate detection of real medication versus this endogenous or food source molecules that can also have a drug usage, me and two students manually curated another layer of information for the drug library, which document them based on the exposure source of the chemical, whether it's purely medical or it can also come from endogenous or food resources. So after I have this reference library for drug as well as their metadata, I was able to analyze the breast milk samples within a timely fashion. So as you can see on these Slack messages, so one day at 11:33, well, right before our lunchtime, Shirley came to me and asked me, Nina, do you have any preliminary data on the antidepressants detected in the 2,500 breast milk samples? So I finished my lunch party with my colleagues, and at 12:22, I was able to give her the number that there are about 14% of samples that have antidepressants detected. So Shirley was really happy with the speed that I functioned. She said, awesome, thank you, Nina. But I really want to say that it's actually the drug library that was awesome, that allows me to analyze drug in these breast milk samples within such a timely fashion. However, this is not the end of the story. So after a few days, Shirley came to me with another question. Well, what if the drugs are present in the breast milk as metabolites? This is an especially important topic because different metabolism in different people is related to the efficacy and the toxicity of these drugs, right? So I thought that's a really interesting question. However, it's really hard for us to analyze drug metabolites using untargeted metabolomics simply because the reference spectra for drug metabolites are so hard to collect. It's really hard for us to acquire analytical standards for drug metabolites because they were just not produced or not available. So how could I actually get reference spectra for drug metabolites? So, well, one approach is definitely I can incubate all the 5,000 drugs against the liver or gut cultures and generate their drug metabolites. But instead of spending a lot of time trying to analyze these sample and data, I started to think, well, can we actually utilize data that's already collected in the public domain trying to achieve the same function? So for those of you not familiar, GMPS is not only a spectral library platform, but it's also a repository that hosts public untargeted metabolomics data. The numbers here are a little bit outdated, but right now there are more than 4,000 projects hosted in the GMPS repository documenting more than 2 billion MSMS reference spectra. So in order to capture the potential drug metabolites from these rich public metabolomics data sets, I use a strategy called modified cosine similarity. basically I utilized the similar fragmentation pattern commonly observed between drug and drug metabolites, as demonstrated here for venlafaxine and this venlafaxine. So using this spectral similarity search approach, I look through the entire public MS datasets for spectra that looks similar to a spectra of the drug. And after a lot of filtering and curation, in the end, we were able to capture potential metabolite spectra we were able to capture more than 12,000 potential metabolite drug spectra, and I believe we covered metabolite for more than 1,200 drugs in our final resources. So before I close my presentation, I want to give you a few examples of why measuring drug is actually so important. So I'm using an example from an HIV cohort because a previous postdoc in our lab actually observed that some microbial metabolites highlighted here, N-isolipids, they have higher levels in people with HIV compared with people without HIV. So when we want to check whether these changes was caused by the infection itself or caused by the medication, we utilize the clinical metadata provided to us together with the samples. So we noticed that using the clinical metadata, which documented the samples based on the number of HIV medications they were using, we didn't see any significant effects for these N-isolipids. So basically, the conclusion was that the changes in the microbial metabolites was indeed caused by HIV infection rather than exposure to the HIV medication. However, when we utilize the GMPS drug library to understand the drug exposure in this cohort, we start to notice that in the people with HIV, so notice that this is already a sample from people with HIV only. We actually noticed that they were using different group of HIV medication. So although they both take medication, they both were infected, they were actually using different treatment regimes, basically. So because on-target metabolomics allow us to precisely read out the drug exposure in these people, we further separate these samples into different groups based on the different treatment regime that they were receiving. And we noticed that this time, the N-isolipid level significantly changed between these drug exposure groups. So overall, using on-target metabolomics, we were able to figure out effects from drug exposure that was not observed when we only rely on clinical metadata. So in the spirit of IMPRINT, we went ahead and further analyzed the breast milk sample collected from the milk repository at UCSD, which covers more than 2,400 breast milk samples. So analysis was completed by a previous postdoc in the lab, Dr. Sydney, and we start to use the drug library to understand the medication exposure in these breast milk samples. So here is an overview of all the medications we observed, and we noticed that neurology, psychiatry was definitely the most popular category, followed by antibiotics and antihistamine, which agrees with the medication exposure for this cohort of mothers, basically. And for the neurology psychiatry drug, some of the commonly observed drugs include antidepressants, drug used for pain relief, as well as drug used for epilepsy. So because we observed antidepressants and antibiotics as the most common category of medication in these samples, we start to wondering, well, are they actually changing the milk metabolome? So the analysis is still preliminary, but so far we definitely see significant changes in the metabolome of the mom based on whether we detected antibiotics or antidepressants or not. So this project, the analysis is currently ongoing. We were in the process of trying to figure out what are actually the metabolites that changed based on the exposure to antibiotics and antidepressants, and what are the biological function of these metabolites, what are the indication for the infant health when they were consuming breast milk with these drug exposures in them. So stay tuned, more to come with regard to this really cool data set that we were working on. And here is my acknowledgement slide. I'm not going to go through the details since I'm running out of my time. Thank you so much for your attention.